Okay, so this brings us to the last lecture of this series, which is the rational treatment of cancer. So the idea was here that we've discussed aspects of cell biology that give us a better understanding of some of the pathways and some of the key proteins that are involved in controlling cell growth, and also some of the key players that get mutated in cancers. Okay, so the idea is now, does this information, can it be used to as a flow-on effect to think about a rational treatment of cancer, so rational drug design. Okay, so what do we mean by rational drug design? Well, when you think of some of the more popular anti-cancer drugs that exist currently, they've been around since the late 60s, early 70s. So they came into being before we knew about oncoproteins and tumor suppressors and all of the other things about cell biology. So these older drugs weren't rationally designed. I think the term is serendipitous design. It was designed by chance. It came across somehow. So one of the examples here, as shown here, is the compound. One of the ones I talked about, a serendipitous discovery, was cisplatin. And this character, Barnett Rosenberg, was studying the effect of electric currents on the elongation of bacteria. So I'm not quite sure what he was doing, but the way I think about it is that we all know that we have these electrophoresis tanks, okay, and they've got positive and negative electrodes on them, and we put our DNA gel there, and we fill them up with buffer, and the electrodes are attached here to these platinum electrodes, okay, and they're platinum because they're inert. And in this buffer here, you've got various salts, chlorides and ammonia compounds, and tris buffers and all that kind of stuff. So we're used to this sort of thing. Now, he was somehow looking at the effect of passing electric current through, effectively through bacteria. So he had a system where he was running growing bacteria in a liquid and running an electric current through there, and there was an interaction between the platinum electrode, which is thought to be inert, go figure, and the buffer, and that was leading to the production of cisplatin. So platinum with 2 chlorides and NH3s. So this compound, sort of cisplatin, was being produced unbeknownst to Barnett Rosenberg, as he was studying the effect of the electric current running through the buffer. Turned out the elongation of the bacteria was caused by the cisplatin molecule, and he figured, well, if it stops bacteria from replicating, maybe it'll stop cancer cells from replicating. So he went away and did some experiments, and now cisplatin, even to this day, 30, 40 years later, it's still one of the prime go-to drugs for the treatment of testicular cancer and other solid tumors. Okay, so this was not rational drug design. Okay, and cisplatin itself causes DNA damage, so it itself is a mutagen. So by having treatment with cisplatin, you're exposing yourself to the likelihood of further mutations to your DNA. All right, so short-term gain and long-term possible consequences. Okay, and if that short-term gain is that you live for longer, then good. So the idea is, with rational drug design, you can design drugs for particular cancers that are less harmful to the individual. So we had a look at some of the occurrence of various tumors and the life expectancy with some of these tumors, and there are improvements with some of these life expectancies and things, but a lot of this is caused due to changes in diet and hygiene and early detection, and a lot of these things, rather than an actual molecular understanding of what's happening Okay, so so the idea is then, what is rational drug design, and what are people trying to achieve from rational drug design? So with a rational approach to drug design, you need to think about a whole bunch of things. So if this is the plasma membrane of a cell here, and here's our DNA here, and here are our little proteins and things that are happening in the cell. So these are the processes we want to try and inactivate to stop cell growth. Okay, so the molecule, the drug, well, firstly you need to get it into cells, and that's the big question mark. So there's certain characteristics of the drugs that you have to adhere to, whether it be the hydrophobicity of the compound, the size of the compound. So we discuss in the lectures some of those issues around the molecule itself that they're designing. Okay. The next thing is, well, what sort of shape of drug is it? What sort of type of drug is it? And how do you make a choice? So for some enzymes in the cell, they've got catalytic clefts here. They've got little cavities in them that bind a small molecule, which is a biological molecule. So things like ATP is a small molecule which kinases use to phosphorylate things. There's also steroid hormones. There are transcription factors that bind steroid hormones to drive growth. There's other nucleotides that drive growth. There's lots of small molecules that are known about that play roles in cell biology. So if you can design a molecule that looks like ATP, but it's not ATP, it's an analog of ATP, then you can effectively inactivate the kinase. Okay, and if kinases are important in driving growth, and you can mimic the effect of a small molecule and inactivate the kinase by putting a blocker of some description into the catalytic cavity, then you no longer have the ability of that protein to bind to ATP to transfer the gamma phosphate onto the new protein. All right, so an understanding of the activity of enzymes is important to know how to then design a drug for that. Okay, and we all know that we have X-ray crystallography and NMR structures and stuff. Some proteins, they bind to DNA sequence, but there's lots of binding sites and interaction points between the protein and the DNA sequence. So it's harder to design one small molecule to disrupt a binding surface that's much larger. And also, we have protein-protein interactions, and there's all these points where the proteins specifically bind to each other, and it's harder to disrupt those with small drugs as well, because it's just a larger surface area. Okay, so understanding the properties of the protein are important, and how it works. So this brings us to these concepts of is something druggable, and would you want to drug it in the first place? Is it a good target? So if it's an oncoprotein that's driving growth, or if it's a tumor suppressor. If an oncoprotein is driving growth, then it's a good target, because if you can knock out an oncoprotein, you inactivate the thing that's driving growth. If a tumor suppressor is stopping growth, then why would you knock out something that's stopping growth? It makes no sense, because the whole point of the drug treatment is to try and reduce the growth of the cells. So leave the tumor suppressors alone so that they can halt growth. Okay. Question is, can you design a small drug that's going to enhance the activity of a protein? And I would suggest that that is in the too difficult basket right now. There might be one or two examples where you can that might be the case, but generally speaking, I don't think it's plausible at this stage to enhance the activity of proteins through small molecule interactions. So it's harder to make tumor suppressors better, but it's easier to knock out the function of oncoproteins. And the example we talked about earlier was, for instance, if the oncoprotein has a kinase domain, you can replace the ATP with a small molecule, then that kinase can no longer function. So there's all those sorts of things. The thing with RAS has got it's got a small pocket that binds a guanine nucleotide. Okay, the GDP, the GTP. So technically, you could design molecules to try and interfere with its function. But the general consensus is that it's not a good target because it tends to drive growth, and you don't want to lock these growth switches on. Okay, so you need to know, is it a good target? And oncoproteins make good targets. Tumor suppressors don't make good targets. And that idea of a good target is based on the function of the protein and whether it makes sense to put all this time and effort in to target it. Okay. Once you decide that something is a good target, the question then, is it druggable? Is it druggable? So we've got a whole bunch of oncoproteins which probably are druggable. There are other functions in the cell which may not be druggable even though they're good targets. So it's a good target, but it hasn't got a catalytic cleft or something. So it's harder to design something to attack it. Okay. There's also which stage in the cancer progression are you trying to target these things? Like DNA repair enzymes. If they've been knocked out, then there's going to be lots of mutations. If you can activate DNA repair, it's probably too late. You can't repair a lot of damage that's done. So there's also the rationale of where you are in the cancer cycle about which sort of things you want to target. So we had those sort of discussions, or at least I went through some of those sorts of issues about druggability and good targets. We also talked about this concept of the fact that a lot of anti-cancer drugs do cause problems in normal cells as well, because it's very hard to distinguish normal cell biology from tumor cell biology. Okay, so that's the perennial problem with drug design. We talked about this idea of we've talked about this earlier in the lectures. We talked about the different kinds of proteins which often occur in different cancers. So there may be so for instance, if you're looking at a population that's that's thought to be, for instance, breast cancer. So you've got lots of a population of a cohort of people who have breast cancer. If you think of breast cancer as a single thing, then when you're designing a drug for that, you might get sort of a low response rate. So it's kind of, you're not quite sure whether it's working or not. If you can break this large population up into subpopulations, and by subpopulations I'm thinking subtypes, people who have the similar cancers. So this is type 1, type 2 and type 3. So the biopsies from these patients are more similar. So it's more likely that these people have a kinase that's been inactivated, for argument's sake, a particular kinase. Whereas in this population, it was scattered. So if you treat the same drug here to this population, you might get a high response rate, which means it's a really good drug. But that high response rate was masked by these populations in here. So 1, 2, and 3. Because these are not responsive, it masks the fact that this population was responsive. So you sort of go, I'm not quite sure whether that drug is effective or not. Where if you can subtype that into smaller categories and have an understanding of what's affecting that population, then when you do your drug treatment here, the same drug, then you get a much higher response rate, and that's much more that's going to drive the process through clinical trials and all the expense to actually design a drug, because it's very effective, as opposed to something that appeared to be apparently not very effective. Okay, but the same drug is apparently more effective if you can look at a subtype that's more homogeneous. Okay, so how do you look at whether cancer biopsies are similar to each other or distinct in terms of trying to work out these categories that might exist? And modern technologies such as gene expression arrays are used for that kind of thing. I don't want to get too much into the technique, but I guess what's going on here is that you've got 2 cells, and within those cells you've got gene expression occurring, and that's what microarrays are measuring. They're a measure of gene expression. So you're looking not at the genome content, but at what genes are activated. So we're looking for messenger RNAs. Okay, so if a particular protein is being really highly expressed in a cancer cell, and it's high, but it's lowly expressed in a normal cell, then maybe that expression is bad. Okay, and you want to know that, and you want to start saying, well, here's a subtype of cancers which have got these genes expressed highly. So the microarray, it works by you those messenger RNAs are labeled to make them fluorescent, and you convert them to DNA, so it becomes sort of a DNA with a fluorescent tag on it. Okay, and then you do the same experiment for the normal cells, and you label up all of the messenger RNAs. So all of the messenger RNAs, might be thousands of them, different types of messenger RNAs, they're all labeled from both cell types. So you're getting these cDNAs, and these are labeled with a blue fluorescent tag, and these are a green fluorescent tag, or sorry, a red fluorescent tag and a green fluorescent tag. All right. You then get some sort of substrate here, some sort of glass slide, and it's got spotted here single-stranded DNA sequences, and each sequence represents the coding region of a gene. And then you simply anneal, you mix these two together and then wash them across the slide. Okay, and then so you wash the slide with a mixture of these fluorescently tagged cDNAs, which are direct copies of the messenger RNAs from each cell type. Okay, and if a particular gene, we know that to be the RAS gene, for argument's sake, if it was if these were colored green, and it's the cancer cell, so if this spot was green, then it would say that that gene is highly expressed here. Okay, whereas if this spot here, which might be another protein, it was red, so if this was red, it would say, well, that gene was more expressed because there was more RNA in the normal cell. So looking at the color profile of the dots, each dot refers to a gene in the genome, you can look at the expression level, and that's really important information. Gene expression is a good predictor of whether you've got protein levels of that in the cell. Okay, and if something is orange, so it's a mixture of green and red, give you this orange color. If it's orange, then there's no difference between the two experimental samples. All right, so that's what the gene arrays are. They're just these techniques that we use to identify levels of gene expression for each gene in typically the genome, using these microarray slides. And there's other technologies as well around these days which haven't yet made it into the textbooks. So there's an example here where they looked at these lymphoma samples, so B-cell lymphomas, and there's a wide range of different types, and from the different types, so the different types are labeled here. There's 1, 2, 3 types of biopsies of patient categories, if you like, and they can look at whether there's certain genes that are overexpressed or underexpressed in one subtype over another, and then they can choose to treat them with drugs. So you're not treating all of those patients with the drugs, because the drugs would be harmful to some, but the cost-benefit analysis, it's better to take the drug if you have the disease that will respond, rather than taking the drug and having no response. So it's just a way of targeting the drug to the the people who are more likely to be responsive. So if you have a better understanding of the subtypes here, and you can see here for the different populations here that they identified different proteins and genes that were present or being expressed, and the viability or the response of the different samples to the drug were very different, and they were able to get a very high response from one subtype to the drug with lower responses from the other subtypes. So if you can do that to actual patients in the actual clinic, then and you can subtype what they've got, then there's a much better chance you can treat them with a drug that's going to be effective and spare them having to be treated with a drug that's ineffective. Okay, so we went through some examples of drugs that have been used in the clinic. There are some successful drugs. How do they work generally? There's a bunch of drugs that have the ability to make cancer cells differentiate. I think all-trans retinoic acid was one of those. And there are some other drugs which are able to allow to make apoptosis happen again in the cells. So they can induce some cell death in cancer cells. So there's a couple of examples given in the lectures of these, which you can quickly have a look at if you're interested. And then we start to look at what I've already talked about, the what sort of things do you want to target in the cells? Are they druggable? Are they good targets? And we went through those discussions. What is druggable and what is a good target are some oncoproteins, particularly oncoproteins that have kinase domains. So there's a little figure here showing a bunch of different proteins which can be targeted in signaling pathways. And there were these compounds that can bind to the ligand binding domain on the outside of the cell. There are drugs that can bind to the tyrosine kinase domains in these tyrosine kinases, and there are some others as well. Okay, so this is an example of a tyrosine kinase inhibitor. So it doesn't look that way by eye, but somehow this molecule is binding to is mimicking the ATP molecule, and it's binding to the ATP pocket and precluding ATP from being used by the kinase to phosphorylate the target protein. So we talked about druggability. Again, here's showing a small molecule within a catalytic cleft of one of these kinases, and I was saying earlier that transcription factors don't make good targets because they have a large surface area where they bind to DNA, but some transcription factors actually bind directly to steroid hormones. And we know steroid hormones are small molecules that can get into cells, so you can design something that mimics steroid hormones. It may also work as drugs. And there's all these examples here of three-dimensional structures of proteins with small molecules shown to interact and affect their functionality. There's also a bit of a discussion about the human genome, and I noticed there was a bit of confusion about this in some of the posters. What this is saying, this little diagram here, it's saying that if you look at the similarity between different tyrosine kinases, you can group them according to similarity. Okay, so you've got these tyrosine kinases here, and there are other kinases. There are, I assume that's calmodulin activated kinases or calcium activated kinases, and a bunch of other types of kinases, not just receptor tyrosine kinases, serine threonine kinases. So it's looking at how similar the catalytic domain of the pocket is of the different kinases and trying to predict, well, if we know that, then it's likely that these are the kinases that will bind to this particular molecule. And again, I guess then you want to start thinking about how you can design the drug to be more effective towards the particular kinase that you want to inactivate, and try and reduce the non-specific effects, because you don't want to perturb too many cellular processes when you're targeting these molecules. And don't forget, these molecules are expressed in normal cells and cancer cells, but if they're expressed more in cancer cells, or if they're more active in cancer cells, then the logic is that the drugs have more effect in cancer cells. So any ways to target these drugs to cancer cells over normal cells is always advantageous. And we also talked about whether cell lines make good models for humans. There tends to be more mutations in cell lines than there is in humans, and also the idea that we all have different sets of mutations makes the issue a bit more difficult. So not only is the question, do model organisms represent the human condition? The question is also, well, do tissue culture, which is human cells growing in culture, do they also provide good models for human cells? And there are some examples where even tissue culture models themselves don't behave as the same as a human body would. And there's lots of reasons for that. And there's also another complication mentioned, is that some cancer cells, they tend to express these normal proteins which are effectively able to pump the drugs out of cells. So these small molecules tend to be pumped back out of the cells. So even though they're effective whilst in the cell, they don't stay in the cell long enough, and then they're pumped out, and therefore it's difficult to get the drug present inside the cell. And then again, it's just looking at some of these different kinases and as targets to some of the successful drugs that have been designed, such as Gleevec and some other ones. And then just lastly, I guess if you go back to the first lecture, remember we're looking at villi, and we're looking at these transient amplified cells which are derived from stem cells. And I included that slide because I knew later on in the lecture series there was this discussion in the textbook about some drugs are effective against the transiently amplified cells, but the cancer stem cells are inaccessible to the drug. So whilst you initially have a good response rate in killing off these transient amplified cells, after a period of months, these stem cells can be reactivated to then produce more of the cells that contain the mutation, because the mutation is present in the cancer stem cells. Okay, and it's just copied into the transient amplified cells. So there's a bit of complication in the literature that sometimes you can kill the cancer stem cells, which will stop producing the transient amplified cells. So you don't see much of a change initially, but over time these will die off. So it doesn't look like it was effective, but it actually was. And the converse situation would be, well, you kill off these transient amplified cells, but because the stem cells are inaccessible, then these stem cells will produce more cancer cells later on down the track, so you get the cancer coming back. So there's lots of complications with drug design, and we're just it's worthwhile discussing some of those in relation to cancer subtypes, where the cells are in the body, and the rational drug design. Do you want to drug it, and is it druggable? Okay, so there's lots of issues there. Okay, well, hopefully that's some food for thought. Hopefully quickly working through the lectures as I have has been beneficial to you. Like I said, I don't think listening to this lecture in isolation will be particularly good as a study tool, but once you've been through the actual course content, I think if you go through this lecture, you should be able to listen to what I'm saying, go, oh yeah, I remember that, I know what he's talking about, oh yes, that makes sense to me. And if it doesn't make sense, then you go, oh, well actually maybe I don't understand the lecture content, maybe I should go back to the lecture content and apply some more revision to that area. Okay, so there's only 5 lectures, it's 10 hours of content. It's not too much to take on board, so good luck with processing that, of working through that content. Good luck. All right, thank you, bye.